Welcome to the FAIRe eDNA session. My name is Miwa Takahashi. I am a research scientist at CSIRO based in Perth on the country of Whadjuk Noongar people. In this session, I will introduce the FAIRe eDNA initiative, along with the guidelines and the resources that we have developed to support FAIRe eDNA data practices. By the end of this session, you will learn how to generate your own custom FAIRe templates and how to fill them following multiple formatting rules. Most importantly, you will be well equipped to make your own eDNA data FAIRe and become part of our growing FAIRe community. The FAIRe initiative was launched in 2023 at CSIRO with the goal of making eDNA data FAIRe, findable, accessible, interoperable, and reusable. The project quickly expanded into a multidisciplinary international collaboration. This expansion was reflecting the growing recognition of both the values and challenges in making these data FAIRe. At the same time, biodiversity and molecular data infrastructures such as ALA, GBIF, OBIS, and eDNA were already investing significant effort into supporting eDNA data within their systems. Existing standards also provide an important foundation for this work. These included TADWIG, which developed biodiversity data standards called Darwin Core, and Genomic Standard Consortium, or GSC, which developed molecular data standards such as MIXES, minimum information about any X sequence. Journals are also recognizing the same challenges around improving data policies and practices. Sharing data openly has become a normal part of publishing scientific research, but making data truly FAIRe is still much harder. So these database managers, data standard developers, eDNA practitioners, journals, we all came together to develop FAIRe guidelines specifically for eDNA workflows and data. This collaborative effort became the FAIRe initiative, and the result is the FAIRe guidelines and associated resources that I will introduce to you now. One of the major outcomes of this collaboration is the FAIRe guideline paper published in the journal Environmental DNA. The content of the guidelines is also available through this website. The guidelines include detailed descriptions of data components and formats, metadata checklist, and formatting tools and scripts. I will now walk you through the website. If possible, please open the website on your own computer so you can follow along. This will help you become familiar with the structure of the website and make it easier for you to locate the information and resources you need when formatting your own data. This is the FAIRe eDNA guideline website. In your browser, you can type in this URL, fair-edna.github.io, or in your search engine, you can type in FAIRe eDNA GitHub, and this page should come up on the top or near the top of your search results. Once you open this page, it will take you to this home, and you can find the rationale and scope of this project. And then at the bottom, you can find this infographic of our FAIRe initiative. I will now walk you through this infographic quickly. Environmental samples are collected and processed through a wide variety of workflows, generating many different types of data and biodiversity information. If the data and metadata are not described in a standardized way, the data sets become difficult to integrate, compare, and reuse across studies. The FAIRe initiative aims to transform this single-use data lifecycle by developing standard formats, formatting tools, policies, and collaborations with cyber infrastructure providers and many other experts. Through these efforts, eDNA data and metadata from multiple studies become interoperable. This also allows eDNA-derived biodiversity data to be combined with global biodiversity datasets generated through other observation methods. And finally, FAIRe eDNA data will enable large-scale reuse and synthesis. Let's go back to this website, and now we're moving on to the guidelines page. This is the largest section on the website and contains a lot of information, so I will guide you through the key sections step by step. At the top of this page, you will find a simplified overview of the FAIRe workflow consisting from step number 1 to number 6. Step number 1 is to generate your templates, your metadata templates, using a script called FAIRe Template Generator or FAIReator. There's also a tool called FAIRe Sheets developed by some of the FAIRe members, Luke Thompson and his team. We will be demonstrating how to use both FAIReator and FAIRe Sheets during this workshop. You then use the templates to record the relevant information from sampling, laboratory workflows, data generation, and data curation processes. Then step number 3 is to validate your metadata using a tool called FAIRe, the FAIRe Metadata Verifier. This helps identify missing mandatory information, formatting errors, and other issues before publication. Step number 4, once your metadata is validated, then you can map your data to meet database-specific required formats using tools like FAIRe to MDT, eDNA to OBIS, and depending on where you want to publish your data. You then publish your data through the repositories such as GBIF, OBIS, or INSDC. These repositories assign persistent identifiers such as DOIs. Then the final step is to provide those DOIs in a scientific paper data statement when your studies are published. There's also a section here on the right-hand side describing where to publish different types of eDNA data. We will not go into this in detail today, as this topic will be covered more extensively during the OBIS session in this workshop series. Multiple formatting tools are mentioned in the FAIRe practice steps, and we will be demonstrating them all during this workshop. The next section after this FAIRe practice workflow is the data formats and components used within FAIRe guidelines. So the first two essential components here are the project metadata and the sample metadata, which describe the project, methods, and sample information. The remaining data components vary depending on the type of eDNA workflow used, for example, metabarcoding workflow or targeted assays such as qPCR workflows. Below this diagram of overview of data types, you'll find the detailed descriptions of each data component and explanation of why sharing them is important for making eDNA data fair and reusable. Then the next figure here, figure 4, illustrates examples of non-curated or raw ASV or TO tables and sequence taxon tables. The key difference between non-curated and curated datasets is that non-curated tables may contain control samples and ASVs from positive control samples, contaminant sequences, or intermediate processing results that are often removed from final curated biodiversity datasets. Retaining and sharing these intermediate or raw data sets can be extremely valuable for data reusers. For example, some users may require more stringent quality control than others, want to understand how the final taxa lists were generated, or wish to repeat or modify the curation process themselves. So providing access to these intermediate data products therefore improve transparency, reproducibility, and in some cases, reusability as well. This next figure illustrates how metadata formats can be slightly different when multiple assays were applied within a project. Once we generate your own custom metadata templates together, you will get a better understanding of these differences, so I will not go into the details now. This section explains the importance of using consistent identifiers and file naming conventions across all metadata tables and data files. For example, project IDs, assay names, sequencing run IDs, and sample identifiers should be used consistently so that separate files and metadata tables can be reliably linked together. Next section here is about the FAIRe metadata checklist. You can download the checklist via this link here, and there are a lot of information packed in the checklist file. We'll get back to this later and take a closer look, but for now I'm going to keep scrolling down this page just so that you can get a better broad overview of the website contents. Here is a data submission and publication section. Available scripts and tools are listed here. Everything is available via GitHub. Example datasets are also available for eDNA studies using both targeted assay and metabarcoding approaches. Then we have frequently asked questions here. Then finally, the useful resources are listed here.